Hey everyone, it's Dr. Marcon. This is part 3 of our cardiovascular physiology lecture series. We talked about the heart, how the heart pumps blood. Now we want to talk about how blood goes to the rest of the body. And we know that we have vessels, specifically blood vessels, that move blood to the different parts of the body. So vessels are conduits that move blood from place to place. And we know that blood flow goes from the arteries, specifically first the largest of the arteries, which is the aorta, coming off of the left ventricle of the heart. So from the arteries to the arterioles, down to the capillaries, and then to the venules, to the veins, and then back to the heart. So blood pressure is highest in the arteries and will decrease continuously as blood flows through the circulatory system. And we're going to go over the different vessels that make up the circulatory system in our body and how blood flows throughout our body. So here we can just see the diagram. We can see the left side of the heart, which will pump blood to the aorta. The aorta will then transport blood to the arteries. Arteries will become smaller arterioles, and then we have exchange of gases and nutrients occurring at the capillaries. We then have absorption in the venules. The venules will then take the less oxygenated or deoxygenated blood up to the veins. The veins will then allow for transport of this blood through the vena cava and back to the right side of the heart. Okay, and then we have again an exchange within the capillary system of the lungs. So we have a reoxygenation of the blood, and we can see that it goes back to that loop. Okay. The first structures we're going to talk about are the arteries. We know that arteries, by definition, take blood away from the heart. We don't really focus on whether or not the blood is oxygenated or deoxygenated, but for example, the pulmonary arteries. Pulmonary arteries are the only arteries in the body that contain a less oxygenated or deoxygenated blood. We say deoxygenated, but really we know that blood that is returning from the body is less saturated with oxygen than blood leaving the heart. So deoxygenated, again, is a bit of a misnomer, but as an anatomy instructor, we often say deoxygenated. But anyways, so arteries take blood away from the heart. Pulmonary arteries take the blood away from the right ventricle and transport this blood to the lung, whereas regular arteries take the oxygenated or the saturated oxygenated blood from the left side of the heart and then transport to the rest of the body. So the first major large structures coming off of the left side or the left ventricle is the aorta, and aorta is the largest artery. So arteries, their structure, they're very muscular and elastic, so they have a large amount of elastic tissue within their layers. They also have a lot of smooth muscle within their layers. So here we can see within the table the different components of the layers of the arteries. So we have, our endothelium, then we have large amounts of elastic tissue, smooth muscle, as well as fibrous tissue, and we can see in this cross section the different components that make up the artery. So functions of the arteries: they take blood away from the heart and help to maintain blood pressure. Arteries are generally high in oxygen, except of course the umbilical arteries and the pulmonary arteries. And they are a low volume vessel, meaning only 10% of the volume of blood is found in our arteries, because the arteries are constantly transporting oxygenated blood to the tissues, to the rest of the body. So very low volume vessel. So the elasticity of the arteries, that characteristic of the arteries, helps preserve blood pressure. Here we can see the blood pressure being preserved during diastole. We know that ventricular contraction is the force that creates the continuous blood flow through the cardiovascular system. As blood is under pressure, it is ejected from the left ventricle, and then the aorta and arteries expand to accommodate it. So here we can see a diagram in which the systolic pressure is stored in the elastic walls for arteries and will be slowly released through elastic recoil, which helps maintain the driving pressure during relaxation. So first we have contraction of the ventricles. The semilunar valve will open and blood is ejected from the ventricles and flows into the arteries. The aorta and the arteries will stretch or expand and store pressure in the elastic walls. Okay, so we can see that this elastic property helps preserve blood pressure, helps preserve that driving force, so that blood is constantly being pushed throughout the arteries and the arterioles. Okay, so it's that elastic property that will preserve blood pressure. So when the ventricles relax and the aortic valve closes, the elastic arterial walls will recoil. And this helps propel blood forward into the smaller arteries and arterioles continuously. So first, we have our isovolemic ventricular relaxation, which will, again, is responsible for decreasing that pressure in the ventricles. With that decreasing pressure, because it is less than the outlet pressure or the pressure in the semilunar valves will now close. So the semilunar valve shuts, which prevents backflow of blood back into the ventricle. And then here we see that elastic recoil of the arteries sending blood forward into the rest of the circulatory system. So as major arteries divide into smaller and smaller arteries, we can see that the wall characteristics change, or the walls of the arteries and arterioles change. There is a drastic difference in composition of these structures. So as they get smaller, the walls become less elastic and more muscular. So here we can see in arterioles, they have less elastic tissue in their walls, but more smooth muscle. The walls of the arterioles contain several layers of smooth muscle that contract and relax under the influence of various chemical signals, and we'll talk about that in a little bit. So arterioles, they are thin and muscular. Their function is basically to partition blood flow. What does that mean? So basically arterioles will direct blood flow to areas that need it the most through changes in their radius, or the radius of the arterioles. So they allow for transport of oxygenated blood to areas that need it most, like tissues and other areas of the body. So arterioles have the smallest total cross-sectional area. This provides the greatest site of vascular resistance. But they don't have the smallest cross-section, and we know that capillaries actually have the smallest cross-section. There is a significant pressure drop before reaching the capillaries, but we know that vascular resistance, eventually we'll learn about how resistance affects flow. Now, the diameter, or the vasomotor tone of arterioles, is highly regulated. We have tonic activity from the sympathetic nervous system. So autonomic nervous system, we have only sympathetic influences. We also have local chemicals. For example, we have hormonal influences that can cause constriction, such as epinephrine, and local chemicals can cause dilation to increase blood flow, especially during exercise. For example, we have autoregulation locally by nitric oxide. So systemic regulation by hormones causes vasoconstriction, whereas local chemicals causes dilation. Again, an example of a local chemical that can cause dilation is nitric oxide, and this is especially during exercise. Here we see an image of a capillary bed and the arterioles that will feed into it, as well as the venules that drain away from the capillary bed. So again, we said that arterioles have walls made up of smooth muscle and can direct blood into the capillary bed. Again, it has that partition characteristic, meaning it directs blood flow to areas that need it. Now, we also have structures called precapillary sphincters. These precapillary sphincters can close off capillaries in response to local signals. Also, when the sphincters relax, blood flows through all the capillaries in the bed and allows for exchange of nutrients, exchange of gases. Now, if these precapillary sphincters constrict, what can happen is blood flow can bypass the capillaries completely and flow through the medarterioles. So here's a medarteriole, or through a arteriovenous bypass, which can send blood directly into the venous circulation. Okay, so we have sphincters that will close off the capillaries, preventing exchange, and instead we have flow of blood from the arterial end to the venule end directly. Okay, again, bypassing the capillary bed. Now we're going to talk about capillaries. Capillaries are the sites of cellular exchange. And we can see that the structure of capillaries, they're made up of a simple squamous epithelium, which is very advantageous for exchange. So they are a thin-walled vessel made up of a simple squamous epithelium, again a thin single-cell layer. Function of the capillaries is for exchange, so exchange between the plasma and the interstitial fluids. So fluids within capillaries is called plasma. Once it is transferred into the interstitium, it's called interstitial fluid. So capillaries are responsible for the exchange between the plasma and the interstitial fluid. Now, this exchange takes place either by movement between the endothelial cells through fenestration or pores, or by movement through the cells. This is called transcytosis. So junctions or fenestrations between the endothelial cells allow for variable leakiness. And again, exchange can occur through cells called transcytosis. And here we see vesicles going through the cells and then being released into the interstitium. The capillaries are narrow, but they are numerous. So capillaries are about 1 red blood cell in diameter. When we saw in the lab, for example, with the fishtail, we saw small capillaries and red blood cells moving through these capillaries in single file. However, despite being narrow, we know that we have a lot of capillaries within our bodies and can make up for about 50,000 miles of these vessels. So the capillaries have the largest cross-sectional area of all vessels. This graph just shows a comparison between the total cross-sectional area of blood vessels and the velocity or speed of blood flow in the circulatory system. So we know that capillaries have the largest cross-sectional area. This is actually pretty advantageous because it allows blood to move slower. We want blood moving slower in these areas to allow time for diffusion. So think of a stream widening, so a stream of a river widening within a rapids to actually slow down flow. Now, even though a single capillary has a very tiny diameter, we said that it allows for just maybe 1 red blood cell to flow in single file. But when you put all these capillaries together, their summed diameters cover an area much larger than the total cross-sectional areas of all the arteries and veins combined. Now, the book gives a really cool example. It gives an example of a package of spaghetti. So we know that each, noodle of spaghetti has a very small diameter, but if you gather many pieces of spaghetti in your hands, the total area occupied by the end of the spaghetti that you see is quite large. Okay, so again, velocity of blood flow is slowest in the capillaries because the total cross-sectional area of the capillaries is quite large. And again, that low velocity flow through capillaries is a very useful characteristic that allows enough time for diffusion. Okay. So here is a picture of the different forces that act on the capillary that either encourage filtration or absorption. So we have a capillary, we have an arterial end, and then we have a venule end. Okay. So another form of capillary exchange is bulk flow into and out of the capillary. Bulk flow is the mass movement of fluid as the result of certain forces. So we have hydrostatic forces or hydrostatic pressure, and we also have osmotic pressure or osmotic forces, which create gradients. Absorption is the bulk flow or fluid movement into the capillary. Filtration is the bulk flow or fluid movement out of the capillary. And we have different pressures again or forces that will lead to either filtration from the capillary to the interstitial fluid or absorption from the interstitial fluid to the capillary. And again, these two forces that regulate bulk flow in the capillaries, we have first hydrostatic pressure. Okay, again, hydrostatic pressure, this pressure is best actually estimated by blood pressure, or mean arterial pressure. This is the pressure that is exerted equally in all directions, will push fluid from the capillary to the interstitial fluid. Okay, so an idea of hydrostatic pressure, again, it is estimated by blood pressure or mean arterial pressure, which encourages filtration, movement of fluid from the capillary to the interstitial fluid. And here on this diagram, we see it represented by PCAP. So PCAP is capillary hydrostatic pressure. Again, this pressure kind of pushing fluid out from the capillary into the interstitium. The next important force that helps regulate bulk flow is osmotic pressure. Now, osmotic pressure is based on osmosis and is based on the number of dissolved particles. And the dissolved particles we're talking about actually within the capillaries are due to proteins. So osmotic pressure is also called the colloid osmotic pressure. So the colloid osmotic pressure, this is the pressure that pulls fluid in or sucks fluid in. Okay, and here we see that the colloid osmotic pressure is represented by pi. Okay, so the main solute difference between plasma in the capillaries and interstitial fluid is due to proteins. Now, proteins are mostly present in the plasma, but mostly absent from the interstitial fluid. And these two sets of opposing forces are called Starling's forces. Okay, and this is what we were talking about in lab. These are the forces that are either encouraging filtration or encouraging net absorption. And we can see on the arterial end, we want filtration of fluid into the interstitium because it's this fluid that will be delivering the nutrients and the oxygen to the interstitium. So on the arterial end, we see that the hydrostatic pressure, the capillary hydrostatic pressure, is greater than the colloid osmotic pressure. And if it is greater than this pressure, than the colloid osmotic pressure, it will encourage net filtration, so fluid out of the capillary. Then we get to a point where it is equal, where there's no net filtration or absorption, and then we get to the venous end or the venular end, where at this point there is more plasma proteins within the capillary, which encourages absorption. So we have an increase in colloid osmotic pressure, and it's greater than the capillary hydrostatic pressure, and overcomes this force so that it encourages absorption of the interstitial fluid. Okay, and again, these are Starling's forces. So from the capillaries, blood flows into small vessels called venules. Although they are similar to capillaries in structure, for example, they have a thin exchange epithelium and very little connective tissue, they are different or distinguished from capillaries by their convergent pattern of flow. So rather than branching off, they converge to form larger vessels. So we know that venules will eventually converge and drain into veins. So we're kind of categorizing venules and veins in this slide because we're talking about their similar structures and functions. So for example, we know that the venules and veins, structurally wise, they have a high capacitance. What does that mean? That means that they can hold a large volume of blood. And they are floppy because they are thinner walled and can expand more easily in order to accommodate more volumes of blood. So they fill very easily and have valves to ensure one-way flow and prevent the backflow of blood. Functions of our venules and veins are to, of course, return blood to the heart. This blood, of course, being less oxygen saturated. Also, another function is to dissipate heat. Our veins actually lie closer to the surface of the body than arteries. So because of the close proximity to the skin, helps dissipate heat, helps kind of cool off our body. Another function, of course, is the storage of blood. So our veins are known as the volume reservoir of the circulatory system. About 60% of the blood is stored in the veins. And we know that veins are more numerous and have a larger diameter than arteries. So with regards to functional characteristics, we know that veins can undergo either a venoconstriction or venodilation. With constriction, this is to decrease the diameter of vein, decrease the blood storage, and then when we do this, we increase the venous return to the heart. Venodilation. This will increase the diameter of the vein, thus decreasing venous return to the heart and increasing blood storage. Okay, and we have certain pharmaceuticals which encourage, either venoconstriction or venodilation, especially in terms of regulating blood pressure. We also have skeletal muscle that actually aids in the venous return to the heart. So the skeletal muscle pump acts by compressing the veins, forcing the blood back toward the heart. So here we see when the skeletal muscle compresses, here we see the gastrocnemius muscle. When the skeletal muscle compresses the veins, they force the blood towards the heart, and this is known as the skeletal muscle pump. So during exercise, our muscles are helping to return deoxygenated blood or less oxygen saturated blood back to the heart. That's why, if you guys are studying for long periods of time, it is recommended to step away from your desk, to get up and get moving, to kind of get the blood flowing and moving. So you need to get some blood moving to your brain to kind of help you study, get some glucose to your brain to help you study. And we also have valves in the veins to, again, help prevent the backflow of blood and encourage more of a forward flow of blood back to the heart. And here is just a summary of the vessels in our body, going from the largest artery, seeing the many different layers of the large artery, which again are advantageous in propelling blood forward and maintaining that blood pressure to propel blood forward into the arterioles and then into the smaller capillaries within the capillary bed, and then allowing blood to move from the capillary bed as soon as we have net absorption from the interstitium to the venules, and then from the venules through the large veins, and then from the veins we can have blood flowing back to the heart via these large veins, the inferior vena cava, superior vena cava. So here again we see that blood flow from the arteries to the arterioles, the capillaries, the venules, and the veins. And this table is great because it explains the characteristics and functions of the different vessels within the circulatory system. Make sure you use this to help you study. So we see different characteristics as well as the functions of the different vessels here. Okay. So we talk about portal systems within the body. Portal system, the definition of a portal system basically has two capillary beds in series, meaning two capillary beds back to back. So basically it has an extra capillary bed. And there are three main portal systems within the body that you guys should know about. The first, of course, being the hepatic portal system. So the hepatic portal vein, part of the digestive system, allows for absorption of nutrients from the gut and filtering by the liver. So we have absorption of our polar molecules that we talked about from our foods, our lipids, our sugars through the monosaccharides, and our amino acids, which are the monomers of our proteins. We know that the liver will process these nutrients and can also use these nutrients for storage. For example, glucose that gets filtered and absorbed also can be stored in the liver as glycogen being that stored form of glucose that we use in states of starvation. So this is one of the main portal systems we have in our body, and we actually can see it here. So we have our capillary beds within the digestive system, which will then drain into either the superior or inferior mesenteric veins, which will then drain into the hepatic portal vein to be filtered by the liver. And then we have another capillary bed occurring within the liver. And here is the hepatic artery, which actually provides oxygenated blood to the liver. So oxygen is delivered to the liver by the hepatic artery. The hepatic artery being a branch of the celiac trunk, one of those first structures off the abdominal aorta. Anywho's, so hepatic portal system, again, having that two capillary bed system in series back to back. Another example of a portal system are the kidneys, specifically the nephrons within the kidneys. The nephrons being the structural functional unit of the kidney, which we'll learn about when we get into renal physiology. So the first set of capillaries, we have the glomerular capillaries. So the glomerulus, anatomically we know, is a tuft of capillaries, and this is where filtration takes place. We filter the blood that flows through the capillaries, and then it gets filtered into that first part of the renal tubule, the proximal convoluted tubule. So that first capillary set being the glomerulus, allowing for filtration. And then the second set, we have the peritubular capillaries that run along the renal tubules of the nephron and are also responsible for exchange. Okay, so again, another example of a portal system within the body. The third portal system we don't really see in this image, but we know that exists, is the hypothalamus pituitary axis. So this is the part of the body. So we know that hormones from the hypothalamus have their effects and travel to the pituitary gland. Specifically, we're talking about the anterior pituitary gland. So, for example, we have releasing hormones or inhibitory hormones such as GHRH, which is growth hormone releasing hormone, or GHIH, which is growth hormone inhibiting hormone, which is released from the hypothalamus to the anterior pituitary gland to either stimulate or release or inhibit release of growth hormone. Okay, that's the first part of the capillary system. And then, the anterior pituitary system also has its own capillary system. So we can see that this is an example of a portal system where two capillary beds are back to back. Okay. We will talk more about the kidneys when we get to renal physiology. So I'm going to stop this section right here, this part right here of cardiovascular physiology. We will then get into the fourth part of cardiovascular physiology, talking about flow. Okay.